Hi, and welcome to this podcast for the University of Alabama at Birmingham's Research Center of Excellence in Arsenicals. My name is Veena Antony, and I'm a program manager at the UAB School of Public Health. Today, we are joined by Dr. Veena Antony. Dr. Antony is a professor in the Division of Pulmonary, Allergy, and Critical Care Medicine at the UAB Heersink School of Medicine. Dr. Antony's research is focused on environmental and translational medicine, cystic fibrosis, pleural disease, and lung injury. Dr. Antony is co-lead over Project 2 for the Research Center of Excellence in Arsenicals, which focuses on understanding the role of bromo and extra-terminal proteins and associated inflammatory pathways in cutaneous arsenical-delayed lung injury. So thank you so much for being here, Dr. Antony, to talk with me about your work with the center.
I'm so glad I could be here.
Perfect. So to get us started, can you tell me a little bit about what the overall goal of the project is that you lead?
Thanks, Veena. I'm happy to do so. You know that exposure to these highly reactive and toxic arsenicals, such as leucite, can cause horrendous skin blistering and inflammation, not only locally, but also in distal organs, such as the lung and the kidney. So my project is on examining what happens to people who survive the acute event of exposure on their skin, so cutaneous exposure to these arsenicals such as leucite, and what happens in the lungs of these people after a period of time. That means if they survive the acute exposure, because so many people die in the chemical exposure to these arsenicals. They have been warfare chemicals for a long period of time since World War I, and because we still have large residual amounts of leucite and other chemicals stored in facilities, and there are accidental and wartime exposures to people, we still need to take extreme care to continue to find methods for mitigating these problems. And my goal in my project is to evaluate the delayed lung injury that occurs in people who've been exposed to arsenicals on their skin. And this can happen after a long period of time, after several months, after they have survived the acute event.
So that leads in really nicely to my next question is about, how arsenicals and specifically leucite really affect the lungs. If someone does survive this exposure and you're looking at, delayed lung injury, what do these chemicals do to the lungs?
So arsenicals come in multiple forms. There are multiple names. It's not just leucite. There's diphenylchloroarsenine. There's another one called diethylchloroarsenine, and another one called diphenylcyanoarsine that can also occur by accidental or deliberate release and use. They can cause significant delayed lung injury, and we developed a mouse model of exposure to cutaneous arsenicals and found that these arsenicals caused acute lung injury as well as delayed lung injury in terms of continued inflammation of the airways and the development of small airways disease, which causes peripheral loss of small airways because the small airways in the breathing tubes get blocked off by the presence of collagen and granular tissue, and this leads to a disease called bronchiolitis obliterans. This is a disease that we have heard of before that occurs in people who've been exposed to burn pits, for example, in the Iran-Iraq wars. This disease causes loss of small airways, and as you know, small airways communicate with the alveoli where gas exchange takes place in the lung. So since gas exchange is such a crucial aspect of our survival and our physiology, for us to get oxygen into the lung, the loss of the small airways because they are closed off can lead to significant loss of oxygen in our blood and significant lung disease. The patient or this person, the survivor, would have increased mucus secretion, increased wheezing, difficulty in breathing, and then low oxygen levels. This disease appears to be, in our mouse model, to be a progressive process. That means it doesn't stop after a period of time. We need to actively do some kind of therapeutic measures to block it. And our data suggests that these proteins called BET proteins, BET signaling and its downstream effectors can be the sources of this continued inflammation. So there is acetylation of histones, and this acetylation of histones leads to further injury and further activation of cells in the local neighborhood where this material has now reached into the lungs and cause continued inflammation leading to lung fibrosis and small airway dysfunction.
And so, that kind of leads into, and you've talked about it some of the research that's going on in your lab with these mouse models. Can you expand on that?
The primary goal of this whole project as part of the CounterACT program from the NIH is to try to find chemicals that can mitigate this problem, mitigate acute lung injury as well as mitigate delayed lung injury. And so we started by using chemicals that would mitigate this disease, and we used JQ1 and also CPI. CPI is another chemical that is available for clinical use. It's used in myelofibrosis, but we were able to repurpose it and use it to inhibit the presence and development of delayed lung injury and the progression of this loss of lung tissue and the important role of the breathing tubes in the lungs, the airways in the lungs, to get oxygen and air into the smaller airways and into the alveoli. We found that using these inhibitors of BET proteins and BET bromo domains protected mice from developing DLI, delayed lung injury. And that was a very important finding. We are at the present time examining the possibility that we could develop in our own drug development core certain chemicals that inhibit both RIP kinase 3 and also BET domains, BRD4. And if you could inhibit both of them together, we have a much stronger inhibitory effect. We are very glad to report that our drug development core does have such a molecule. At the present time, we call it the SRI molecule, and that can help these subjects or the mice in our research to be protected from developing long-term effects of exposure to arsenicals on the skin.
Well, Dr. Antony, this is very exciting research that you're involved in and that you've been doing as part of the CounterACT network. I know we're running out of time shortly, but is there anything else that you would like to share with our listeners?
I think I'd like to comment a couple of other things. One is that, we have not yet, I'll just say that we have just begun to explore the role of neutrophils in the development of these problems. We find that neutrophils, as they go past the skin area where the injury was, get activated. And once they get activated and they transmigrate into the lung, they slow down in the lung tissue. And as they slow down through the endothelium, they're able to extravasate out into the lung tissue itself. And when they do that, then they release all the toxic contents, including oxygen radicals, proteases, that continually cause inflammation in the lung itself. So this is an area and a possible pathway that we are just beginning to explore as a cause for the delayed lung injury. The second point I would like to make is that we as a community and the National Institutes of Health are eager to continue to work in these areas because it is very critical, particularly at this time, this fragile time in the world status, because there are significant amounts of product available in Eastern Europe, and we need to continue to support and find ways to help people who accidentally or by deliberate release during wars are exposed to these arsenicals. So I believe that the NIH recognizes it, and all investigators in the Center for Arsenicals recognize that this is the primary reason we have to continue this work, to find ways to prevent and mitigate the problem once it starts.
I think that's a great kind of segue and to end this. As you said before, this is not a threat of the past, but this is a potential threat of the present. I also want to thank you for all of your work that you have done as part of our center, and all of the researchers within our Research Center of Excellence in Arsenicals, and like you said, everyone who's working in this field, this very important field. Well, Dr. Antony, I want to thank you for being here today. It was great learning more about the lung project of our center and the work that you are doing in developing countermeasures to mitigate arsenical-induced lung damage. I so appreciate you joining me today.
Thank you so much, Veena, and thank you for your support of the center.
Absolutely. And thank you for listening. Please tune in next time for another episode from the UAB Research Center of Excellence in Arsenicals.